Good afternoon, everybody. Hopefully you can hear me and looking forward to welcoming you all to this exciting seminar. We're going to give everyone just a couple of minutes to make sure that their technology is working. So feel free to just make sure you can hear us and see us. And I'll be handing over to Professor Tajipar in a few moments after a short intro. To give people 1 or 2 more minutes just to join. Great. Well, in the interest of time, we will crack on. Welcome everybody to our summer exciting seminar. This is a particularly special one as we are celebrating 30 years of single molecule FRET, 30 years since the report of the first single molecule FRET measurement made by TJ himself, which is why we're super excited to have TJ present to us today. He's one of those true pioneers of single molecule biophysics. His work's had an undoubtable influence on the field over the last 30 years. And, many of those early years at the University of Illinois, and then at Johns Hopkins, and then recently he moved to the Harvard Medical School and Boston's Children's Hospital, where he's leading a research program developing cutting edge methods to answer fundamental questions in biology. So I think it's fair to say most of us here in the room have been influenced by TJ single molecule fluorescence in one way or another, from those foundational advances in single molecule FRET and imaging to more recent work on genome biology, mechanobiology, and CRISPR. His lab has always pushed the boundaries of what's possible. So it's a real privilege to have him with us today. TJ, really excited for your talk. I will yield the floor to you now for that and stop sharing and ask you to share your screen. Thank you. You are currently muted.
Okay. Yeah, thank you, Tim, for the invitation, and I'd like to thank everyone for joining this Monday morning, actually, in the US. And I hope you are enjoying the soccer matches as much as I have. All right, so let me share my screen. All right, so I picked this title, Conformational Control from Single Molecule Threat to Biotechnology, because we can use measurement tools to gain mechanistic understanding of a biological system, and then use that knowledge to develop novel biological tools that can be used for many other systems. And I will describe a few examples. But first, I will tell you a little bit about this anniversary, because my first paper as a grad student was published this year in PNAS and reporting single molecule FRET. And I want to give you a bit of a personal story about that before going into the main topic. And then I will end by showing you some unpublished work on prime editing using single molecule FRET. So we know that proteins have atomically precise structures. They also have multiple conformations, as shown in this example, where Helicase enzyme is using ATP, hydrolysis and release to unwind DNA into single strands. And with the advent of AlphaFold and various structural biology tools, we can now easily determine or predict 3D structures of biomolecules and biomolecular complexes, and even see multiple conformations. But it is still very difficult to actually piece them together into functionally relevant conformational transitions. And it's really difficult to measure the kinetics of conformational changes just from static snapshots, and also understand which of the many steps is rate limiting functionally. So we believe that single molecule FRET is really providing a novel and powerful complementary approach to relate structural changes to function, as shown in this illustration made by Harold Kim, using I think one of the most well-known sports players here. So in single molecule FRET, you have two dimolecules, green and red, donor and acceptor. Normally, when you excite the donor molecule, you get green photons out, but if you have a second molecule, red molecule nearby, you can get direct transfer of the energy without emitting actual photon, and then you get red photons instead. So when you have 100 times the donor is excited, how many times you see red photons? That's called energy transfer efficiency. And it is a strong function of distance r between the two molecules. And the relevant length scale is about a few nanometers, distance at which efficiency of energy transfer is about 50%. And if you plot this function as a function of r, you can see that there's a pretty nice regime where the change in distance is actually linear for in terms of FRET efficiency. So you can get down to just a few angstrom scale precision in measuring distance changes. In fact, if you know where these dyes are attached on your biomolecule, you can infer based on relative intensities of green and red emission what the conformations are being sampled. So actually this year is a 30 years anniversary for my first child. So it's a personally very important year, 1996. So that was my first child. But my second child also born in the same year was this paper showing that you can measure FRET from single donor and single acceptor pair. And this was communicated by Nacho Tinoco, late Nacho Tinoco, was the pioneers in single molecule nucleic acid experiments. And this was basically headed by my two colleagues now, boss Shimon Weiss and collaborator Paul Sevin, who are corresponding authors of the paper. It ended with this sentence in the abstract: monitoring conformational changes within single biomolecule may be possible in the future. And I didn't think this was going to happen because this was done on a dry sample. But now many laboratories, due to efforts from many different labs and people, we are now using this tool very routinely for measuring conformational changes in other activities. And here's a nice another nice metaphor that we often use. Just to tell you that this was not planned. So it was really a random walk into single molecule studies for me personally. So I was doing some semiconductor studies as a PhD student, and then Shimon got some money and then told me to read Eric Patrick's PhD dissertation and build a new kind of microscope to study semiconductors. So I did, but it was not useful for semiconductors, at least at room temperature. So then, and Shimon said that, okay, see what Eric Patrick, Pat Ambrose, and Sonny did using this microscope to see single fluorescent molecules at room temperature. So let's do that. And I said, but just doing single molecule is not new anymore. So how then can I get a PhD? So then we say, okay, let's do two molecules, and that became a single molecule thread. But how do you put two molecules close to each other? And Paul Sabin taught us how to do it. And besides, he also told us that this can be useful for biophysics. So when I was a postdoc, I was looking for some biological systems to study using this method. And I encountered this review paper by Tim Norman. And it actually talked about an enzyme that I didn't know about. It's a DNA helicase that uses ATP to move along the DNA to separate DNA strands into single strands. So that's the unwinding enzyme. And we've been studying this class of enzymes for now maybe 25 plus years. Another powerful single molecule technology is optical tweezers. And we decided to combine FRET with optical tweezers into a single microscope so that we can measure conformational changes using fluorescence as a function of force applied. But we can also use optical tweezers alone to measure molecular activities by changes in distance between the tethering point and the optically trapped bead. So here's an example. So it's a helicase called UVLD, and it unwinds DNA, but it's a poor unwinder by itself. It has two different conformations, open and closed. And what was not clear in the past was which of the two is a functional form for DNA unwinding. And so we can actually label two cysteines engineered into the helicase with a random mixture of green and red dye molecules so that high FRET reports on the closed form and low FRET reports on the open form. And then we built a microscope that combines high-resolution optical tweezers with confocal single-molecule FRET, so you can measure DNA unwinding using optical tweezers, unwinding of this DNA hairpin, and measure FRET from a single enzyme to measure its conformations at the same time. And here's a tabulation of many events. And what you can see here is that for the events where you have high FRET efficiency on the x-axis, unwinding velocity is positive, meaning that in the closed form, enzyme is unwinding the DNA. But when FRET is low, then velocity is negative, meaning that enzyme is actually going backward to allow the DNA to reanneal in its wake. So this means that these two conformations are not just random conformations, they're actually critical in regulating the unwinding activity. So one idea we had was, what if you use cross-linking between the two cysteines to staple this enzyme into the unwinding active closed conformation? And that's the work done by, and that will also prevent the open form needed to go back. And this was done by a grad student, Sinan. And then when he did this using the replicase, here he calls it repx for X for cross-linking, and apply this optical tweezers unwinding assay. And he was able to show that this cross-linked enzyme can unwind DNA processively over thousands of base pairs without falling off, even against high applied force. By itself, it has barely detectable unwinding activity. So by doing this stapling into the unwinding active form, you can convert a minimally active enzyme to a super helicase. So that was a really exciting discovery using the mechanistic understanding to change enzymes' behavior. We've been then using this super helicase for various different applications. Let me show you just two examples. First is isothermal DNA amplification. So in this work, we developed what we call a SHARP for SSB helicase-assisted rapid PCR. And that's an isothermal DNA amplification method using super helicase. So here, we don't actually do thermal cycling as in PCR, we use a super helicase, in this case PCR-M6 helicase, to unwind the DNA. And then this allows us to load the primer onto the beginning. And then SSB protects the unwound DNA strand. And then DNA polymerase will copy that DNA over. And this process can continue at a constant temperature. So this work has been published. And in an unpublished work, there's a preprint here. We have also shown that unlike PCR that struggles in amplifying repetitive sequences, you can see that just 5 repeats of this particular sequence we are using really makes it challenging to amplify using conventional PCR. But if you use our method SHARP, even 35 repeats can be amplified without any detectable side product. So this is an example of it using super helicase for interesting biological applications. Let me give you another example of using this for genome imaging. And here we realize that the conventional way of doing fluorescence genome imaging using DNAFISH for fish for fluorescence in situ hybridization actually, although it's powerful, it has some limitations. So here you need to label your probes and then denature the genomic DNA inside the fixed cell by going to very high temperature so that the probe can bind to the target region in the genome. And you can image this using a microscope. But because of global denaturization, we can have a lot of off-target binding by the probes and requires harsh conditions for treatment. And this cannot be used to detect single nucleotide polymorphism because this annealing process is not really sensitive to single point mutations. So we developed a method that we call GOLDFISH, genome oligopaint via local denaturation fish, where we use our Cas9 nickase to make a nick at the location of interest for the genome. And then we use a super helicase to unwind the DNA starting from the nick. And then generated a single-strand DNA region can be targeted using fluorescent labeled fish probes. So we can actually decorate, for example, 2 copies of the same gene in 2 chromosomes within the same cell. So this works now really, really well. And let me show you just one example of applying this to the tissue setting. So here you're looking at human breast cancer tissue, but this cancer tissue is known to have amplification of this HER2 gene. And if you do our GOLDFISH method onto this section tissue sample, you can see that for each of the cells within the tumor region of the tissue, there are a lot of these spots due to GOLDFISH due to HER2 gene amplification. And in fact, you can also see HER2 protein immunofluorescence decorating the membrane of all these tumor cells. But if you look at the normal part of the same tissue, then each cell shows you only two dots, showing that we have the really good sensitivity to detect every copy of the gene and then quantify HER2 amplification level even in the tissue setting. So that's another example of using the super helicase for biotechnological applications. So this is one cycle of example where we learn about different protein conformations and what they're good for using single molecule FRET. And then we use that understanding to develop new technologies through conformational control. Here's another example. As you know, CRISPR-Cas9 is a powerful method for genome editing. And you can think of it as a pair of scissors that can be targeted to anywhere you want in the genome using a guide RNA. one limitation has been that it's a pair of procedures that are not synchronous. So we don't normally know exactly when DNA cleavage occurs. And as a result, it's hard to study DNA repair that ensues or genome editing process, especially early steps. So we developed a technology that we call a very fast CRISPR, where we allow Cas9 to bind the target DNA, but not cut until light removes the lock. And then pre-bound Cas9 can close and cut the DNA within seconds. And then you can use it as a beginning of DNA repair or genome editing process that you can study in fine details. So how does it work? Let me get into a bit more details about Cas9 mechanisms. So Cas9 is a protein that brings this guide RNA, and that red region actually is a guide RNA portion that is complementary to the target DNA. And that's the target DNA is a region called PAM that is recognized by the protein itself. And then this binding to the PAM sequence will result in unwinding of just few base pairs next to the PAM region. And that base pair against the guide RNA. And if the matching is good, then this unwinding of the target DNA and annealing to the guide RNA continues until you reach a critical number, about 17 base pairs of match. Then the Cas9 will undergo conformational changes to bring the nucleases to the cleavage site. It will make 2 cuts and to create a DSB for double-strand DNA break. And then the cell will take care of the rest, giving you ideally your desired outcome of genome editing. We also knew from various studies that you don't need as many base pairs to have a Cas9 binding to the target DNA stably. You only need about maybe 9 to 10 PAM proximal matches for that. So let me actually show you our own data to confirm and support these statements here. So we previously used single molecule FRET to measure Cas9 binding to target DNA. So the assay is shown here. We have green labeled DNA on the surface. The red is the 20 base pair region that is matching the target guide RNA sequence. And then if you add red labeled Cas9 guide RNA complex to the chamber, it'll bind and then unwind the target DNA strand, and then you'll get a high FRET efficiency between the green and red molecules upon formation of this nice complex. So here's a single molecule FRET histogram of such a reaction. When you have 20 base pairs matched between the guide RNA and target DNA, we see this nice high FRET population shown here, and this is the donor-only population. But if you use a control DNA, where DNA sequence does not match the guide RNA, non-cognate DNA, we don't see that high FRET population. If you use just the RNA alone without any Cas9, then you don't see that either. So using this assay now, we have done a systematic analysis. This is a bit busy, but basically going from no base pairing, non-cognate between the guide RNA and target DNA, all the way to complete base pairing at 20 base pairs. As you increase the number of base pairing match from the PAM proximal side, we can see at 7, 8, 9, 10 base pairs, you see this bound state appearing. And it doesn't really increase further as you increase the number of matches between the two strands of DNA and RNA. Therefore, this also shows that you actually need about maybe 9 to 10 PAM proximal matches to allow stable binding of the guide, the Cas9 to the target. What about the second statement? Cleavage competent conformation requires more than 16 base pair match. And we actually use DNA unwinding as a proxy for competent conformation acquisition. So in this case, we label the DNA with a green and red dye molecules so that initially you get high FRET, but as it unwinds, then you see a reduction in FRET. In fact, you can see here that when you have fully matching the guide target DNA, then we see this unwound low FRET population showing up. And as you remove the number of base pairing matches to having now only 16 base pair matches, then the unwound fraction is almost entirely gone. So it looks like the DNA unwinding required for cleavage requires more than 16 base pairs of match, whereas a binding requires only about 10 plus base pairs of match. So there's a window of opportunity that we are utilizing. So by actually inserting this photocleavable occasion groups in the palm distal segment of the guide RNA, you can actually stop it in the bound state. And then when you're ready, you shine light to remove the blockades. And then reaction will then continue to complete annealing between the guide and the target DNA strand. Then you get cleavage within seconds. Then you can study what happens afterward. So if you want, all of the Cas9 molecules are bound to the DNA, waiting for your signal that you deliver using light, and then they will synchronously start the reaction that you can study. And it works really beautifully in living cells. So here we are measuring cleavage fraction, double-strand DNA break fraction as a function of time after shining light. And even at the earliest time point of 30 seconds after shining light, we see half the DNA molecule has been already cleaved. And this is much, much faster, several hundredfold faster than the previous record using chemically inducible Cas9. So we've been using this now to study DNA repair of various kinds. And so here's one high payout. So by doing synchronous cleavage, we can measure repair from a shared time zero. And in fact, this paper published 3 months ago didn't use any single molecule experiments. It didn't even use any imaging, except that Illumina sequencing machine has imaging platform. And we were able to make a nice model of how Ref51 filament can do homologous search during double-strand DNA break repair, and using the scanning by motor protein called cohesin. So that's another example. Let me move to the final section of the talk about prime editing. So that's unpublished work. And so prime editing is actually maybe next generation of genome editing beyond Cas9 generating double-strand DNA break. And it is a very versatile tool without generating potentially toxic double-strand DNA breaks, but the challenge is low efficiency in general. So let me tell you about prime editing. So what are the components? First, you need a protein prime editor, which is a fusion between the Cas9 nickase and reverse transcriptase. And then we need to have PEG RNA, that's prime editing guide RNA, PEG RNA. It needs to have target recognition that is standard for any Cas9-like systems. But we also have additional instructions about editing. So that's editing instructions shown in this light blue color. So the way it works is shown here. So you need this targeting instruction to bind the target DNA, and here TS is for target strand and NTS for non-target strand. Once target is found, DNA is unwound, and by annealing the target strand to the spacer or element of the guide RNA. And then this will trigger a nicking reaction of the non-target strand. That's by design. So non-target strand is nicked. And then that non-target strand is annealed to PBS. So that's the instruction portion, that's a primer binding sequence. So now once this annealing occurs, then this NTS functions as a primer for reverse transcription. So that this reverse transcription this enzyme actually then you extend that DNA using this reverse transcript template RNA, RTT, as a template to incorporate your sequence that you desire into this. For example, in this case, you can incorporate single base pair modifications onto the DNA. Once extension is done, then this newly made DNA strand will actually compete with the existing strand. There will be some equilibration between the two. Eventually, this flap is resolved, and in an ideal case, you get the actual editing of changing a base pair at this particular location. So that's the entire cycle. The problem is actually generally low editing efficiency. And so the question is which molecular steps are limiting the efficiency? And we can actually use single molecular FRET to actually study this multi-step reaction in a very clean way. And this is a project led by a postdoc using Kang. All right, so we'll be looking at the reactions in 3 different steps: unwinding, PBS annealing for primer binding sequence, and DNA extension. So for unwinding, you'll be using the same exact single molecular FRET assay we use for Cas9. So before unwinding, you get high FRET efficiency, and after adding prime editor, if it unwinds the target DNA, then it'll give you this low FRET efficiency. So that's the idea. So when we actually purify this prime editor and then made our own pegRNA and then added this to the target DNA for unwinding, we are quite surprised that even after 5 minutes, we couldn't see any unwinding. FRET efficiency stayed high, and it was puzzling. Maybe our enzyme was not active, so there are some issues. Maybe RNA was degraded and so on. So what we did was to increase this prime editor and guide RNA, pegRNA concentration by a factor of 15, and then we were able to see some hints of unwinding this low FRET state after 5 minutes. So we thought, oh, maybe our prime editor is not functional. Maybe it died during our purification. So we instead use standard Cas9 nickase, but with the pegRNA, and still saw this very inefficient unwinding even at 15x concentrations. But when instead, when we changed pegRNA to the regular guide RNA, but using prime editor that we purified, at the normal concentration, we saw rapid unwinding. So clearly, pegRNA is inhibiting DNA unwinding, which we believe is a critical intermediate for the nicking reaction. So you can actually quantify this unwound fraction as a function of time and show that compared to prime editor plus pegRNA, if you replace this by a prime editor with a regular guide RNA, we see much more efficient unwinding. So the question is why? Why does pegRNA inhibit DNA unwinding? This is something that no one told us about, at least in early publications of prime editing. Now, if you look at this organization of the productive state where this nicked strand is annealed to PBS for reverse transcription, you can realize that there's a spacer that is complementary, but there is another one that is complementary and so on. There's a lot of things that are complementary to each other and so on. So then we realize that actually this PBS is complementary to the spacer region of the guide RNA. So they will actually form this auto-inhibited state, hairpin structure that will actually prevent guide RNA to target the DNA structure. In fact, this was realized by a different lab, and they were able to insert some mismatches in this region of the PEG RNA to increase editing efficiency. So that was the hypothesis we had. This is the reason why we don't get unwinding with PEG RNA. So what they did was to actually introduce mismatches to the PEG RNA to see whether it gets better. So we use the same unwinding assay. And if you introduce one mismatch or two mismatches, 1 mm, 2 mm, you can see greatly enhanced DNA unwinding acquisition of the low FRET state even after the first minute. Yes, so indeed, mismatches can restore DNA unwinding even when you use a PEG RNA. We can also quantify this for various different mismatch construct and showing that even a single mismatch can greatly enhance the activity. What about PBS annealing? Can a mismatch can also enhance PBS annealing? So if we now use an FRET assay between a dye on the non-target strand on DNA and the PEG RNA here, initially you expect to see low FRET efficiency, but once PBS annealing occurs, we should see high FRET efficiency. When you do the same experiment with the perfectly matching the PEG RNA, then we see very little annealing, a high FRET state. But if you put two mismatches, you see now annealing going on. So even before annealing, we see some intermediate FRET stage, but high FRET stage can be seen only when you introduce mismatches. So yes, indeed, mismatches can also enhance the second step of PBS annealing. What about DNA extension? To read out the extension, we perform the reverse transcription experiment in addition by adding dNTPs, but we mixed in a small amount of Cy5 labeled DATP so that when it gets incorporated into one of the two positions on the newly made DNA, then you should also see high FRET efficiency. And that also shows that compared to DNA alone, no except red molecule. If you use no mismatch, fully matching PEG RNA, then we don't see any high FRET state or incorporation of that reportion DNA extension by reverse transcriptase. But when you put mismatches, then you see now extension is happening, showing that indeed having mismatches artificially introduced the PEG RNA can actually really accelerate all three of the early steps of DNA unwinding and PBS annealing and DNA extension. Finally, another postdoc based in the lab tested these different strategies inside a cell. So first thing that she did was that she used a regular Cas9 to measure conventional editing efficiency, generating insertion and deletions. And the bottommost one is that we are using PEG RNA and without any mismatch, generally see very reduced efficiency. But if you use mismatch a PEG RNA of two different locations, then we see greatly enhanced activity consistent with the idea that having mismatches can actually lift automation even inside a cell. And it's not as good as regular guide RNA without this PEG element, but still much better than PEG RNA without any mismatch. Now, if you put a mismatch and then read out the prime editing efficiency, if you put the mismatches in the primer binding sequence PBS region, then we see a modest increase in editing efficiency. But if you put the mismatches into this reverse transcription template regions, actually, as you put more and more mismatches, we see reduced final edit, which was a little disappointing. So that improved all this, we can improve all these steps, but you do not always get a better final edit. It depends on where you put these mismatches. And this is how we can rationalize our finding. So if you have mismatches in the RTT, reverse transcriptase template region, then once you make this extension of the DNA, and then this will have to compete with the original DNA through flap equilibration. But in this configuration where you contain the edit in the duplex region, there'll be a lot fewer base pairs. That's energetically unfavorable compared to the other configuration that lacks edit. And as a result, you mainly stay here, and flap resolution will most likely result in the restoration of the original DNA sequence. But if you have mismatch in the other section, it's more likely that you can still keep that edited portion in the duplex during flap equilibration, and you have a higher chance of obtaining the desired edit. So this shows that you can gain early kinetics using the mismatches, but it can be lost during later processing. So the conclusion of this part is that prime editing is conformationally gated, and pegRNA autoelimination by forming this hairpin slows DNA unwinding and subsequent early steps. By applying selective mismatches, you can improve all these steps, but later flap processing can determine the final outcome. So you need to consider both aspects in designing the pegRNA and for improved editing outcome. And finally, single molecule kinetics here explains where efficiency is gained and where it is lost. Right, so that's the end of my talk today. And the message here is that we can learn using single molecule technologies what different protein conformations are good for, their functional roles. And then again, using the knowledge again, you can develop interesting and powerful biotechnologies that utilizes a conformational control, either using light or chemical cross-linking. Thank you.
Absolutely brilliant, TJ. Thank you so much for that. Happy to open the floor for questions now. If you just want to raise your hand and then turn on your mic using the chat settings, then that would be fantastic. And just while we were waiting, I mean, it's great to see the flow from single molecule insight into the biotech space. I think that's really encouraging. One thing that struck me on some of your early work, the mid-FRET peak in the early in the middle story you were talking about, that seemed to shift around a little bit in terms of its position. Is there anything interesting going on in the detail there?
Oh, that's that's a good point. So I think the unwound peak shifts around based on how many base pairs actually you have, likely because the actual number of base pairs determine the FRET value. Yeah, physically. Yeah, so that we see a general shift of that FRET peak position as you change the number of base pairing.
Understood. Brilliant. So if anyone does want to ask a question, under the React button at the bottom of the thing, there is the raise your hand. You'd be very welcome to ask any specific questions that you may have. I'll give people a couple of minutes to think those through. In fact, we have one here in the chat now. So from Martin, do you want to turn your mic on, or do you want to read it out yourself? I'll read out Martin's question, TJ. So Martin, thanks for very nice examples of smFRET application. Question relating to the pegRNA in prime editing. How exactly did you isolate capturing efficiency of individual steps, for example, unwinding, annealing, and extension, to see that all three steps got faster? I would assume only unwinding is elevated with mismatches and not the other. Would you also see increases in FRET of the subsequent annealing and extension because you removed the bottleneck, or did I understand it wrong?
Yeah, that's a good point. So we are measuring these three different steps separately. We're not measuring them simultaneously. And my guess is that what we are removing is the first bottleneck of DNA unwinding. But our data also shows that once you remove the bottleneck, the two subsequent steps do not act as an additional bottleneck. For example, you may nick the DNA, but maybe annealing between one DNA strand and the pegRNA, could be a lot slower than the initial unwinding, but we found that not to be the case. So that was something that we didn't necessarily anticipate.
Brilliant. Torsten.
Yeah, hey TJ, thank you very much for sharing this insights into the molecular insights into how nucleic acids can be manipulated by enzymes. So what I was wondering is, do you see effects of dyes on the enzyme efficiencies? Do you have to play with the dyes, or is the side-dyes just working perfectly there on the base pairs? On the base?
Yeah, I think generally they work fine. So in many cases we can do conventional biochemical assays with and without the dyes, and when you can measure the just reaction outcome after maybe 30 minutes, we see the same. But sometimes you can also even measure the kinetics, and we see maybe little effect. Maybe the kinetics do not change within a factor of 2. But that I guess needs to be checked individually. And we certainly do not want to put dyes where we expect to see a strong interaction with the protein.
Yeah, sure. Okay, thanks.
Any more for any more? In which case, I would like to thank TJ again for a fantastic seminar with us here, but also TJ for all your work over the last 30 years. That you've really driven this field forward in a fantastic direction, and I know you've had an influence on many of us in the room here and elsewhere. So thank you again for your time today and your ongoing efforts in this space. And we at Exciting Instruments look forward to supporting you and others throughout the next 30 years as we approach that great milestone. Thank you all for your time, and look forward to talking with many of you over the coming weeks and months at various places around the world. Thank you. See you
All right,
soon.
thank you so much. Thanks everyone for joining.